Many thanks to the organizer for letting me talk today about my, what is my presentation? About my research topics. As Carmen said, I pretty much deal with rare diseases, and in particular with lysosomal storage diseases. Okay. But today I'm going to talk about Fabry disease, which is one of the most frequent lysosomal storage disease. Fabry disease is a progressive multisystemic disease due to an impairment of alpha-galactosidase A, which is which has to hydrolyze glycosphingolipids in the lysosome of the cell. When the enzyme is impaired, the glycosphingolipids are accumulated into the cells, eventually causing a damage in the tissue. The most abundant products that are accumulated in Fabry disease in particular are the GB3 and the lyso-GB3. The gene that encodes for the alpha-galactosidase A is mapped on the GLA, on the X chromosome, so the inheritance pattern of this disease is, of course, X-linked. Due to the ubiquitary expression of this enzyme, the clinical symptoms of the pathology may be really highly variable, and they can basically affect every important organ or system. So they range since very severe symptoms like transient ischemic attack, ventricular hypertrophy, renal failure, to milder symptoms. But the ones I want to point out are the peripheral pain and the acroparesthesia crisis that this patient experiment during their everyday life, and which are really impairing their quality of life, general quality of life. Due to the variability of the clinical symptoms, these pathologies are really often their diagnosis is delayed. And in a first step could be also, the patient may be also misdiagnosed with another pathology. As a matter of fact, the incidence of the pathology in 1999 was ranged to be about 1 in 117,000 born, but we now know that mutation in the GLA genes may be present in up to 1 in about 4,000 newborn, based at least on the screening performed on asymptomatic newborns. From an histological point of view, we can see how the conformation of the glycosphingolipid deposit is into the cells. They usually appear like a zebra body, which are, which are made up of a lamellar structure of periodic electron-dense and electron-light bands. And these deposits are basically present in every cell type, but they are very common in podocytes and in endothelial cells of blood vessels. The treatment that up to now exists for Fabry disease is the enzyme replacement therapy. This kind of therapy is based on the injection of the recombinant enzyme directly into the bloodstream. And the protein is uptaken into the nearest cell through the mannose 6-phosphate receptor. There are currently 2 available drugs commercialized. And in a very recent review published this year, they reported a summary of all the trials performed with the 2 commercial drugs, showing that there is no significant difference between the 2, that they basically reduce the GB3 levels in plasma, and also partial reduction in the kidney and in the heart. But they are pretty much active against peripheral pain crisis. However, this treatment has important limitations. In fact, they have a very low biodisponibility, and indeed, we need to repeat injection every couple of weeks. They have a low capacity to, the protein has a low capacity to be uptaken in other cell types which are not endothelial cells in the blood vessel. And also, they are not active against neurologic symptoms since they cannot cross the blood-brain barrier. Moreover, the production of antibodies against the two proteins have been reported for both drugs. So there are the research field of the new therapeutic strategies is really active in this moment, and there are a bunch of possibilities based on gene therapy, cell-based therapy, ERT, substrate reduction therapy, and also pharmacological chaperones. The pharmacological chaperones are non-covalent inhibitors of the enzyme, in this case of the alpha-galactosidase A, which act as scaffold to allow the correct folding of the mutated protein and made it skip the quality control mechanism of the ER. So this allowed the protein to traffic into the lysosome. Once it reached the lysosome, the inhibitor is released due to the acidic environment of this organelle, and the mutated enzyme can at least partially hydrolyze the accumulated substrate. I'm explaining this because based on the pharmacological chaperone therapy, there is now a new molecule, active compound, that is about probably to enter in the market since it's in clinical trial phase 3, and many trials have been already developed with this molecule. The other chance is to make a strategy based on the transduction of cell with an adeno-associated viral vector, but the attempts up to now are still in the preclinical phase. So we are not sure this can really be used for the therapy of Fabry disease up to now. So what I'm doing at the EB is that we recently got financial support for a project on Fabry disease, and we are focused on 3 main objectives. First of all, we want to facilitate early diagnosis through screening on patients with a clinical suspect of Fabry disease or symptomatology that is of unknown origin. Also, we want to clarify what are the molecular players of the pathophysiological mechanism of Fabry disease, since this may help to find new biomarkers and new therapeutical targets. And finally, we are planning to make up a gene therapy vector for possible treatment of the therapy. Of course, at first attempt, the first objective is the one we have made most effort on this objective and starting to get some preliminary result about the other 2. So what we offer at the EB is a service for diagnosis of Fabry disease, and so we collect a blood sample in form of dried blood spot, and we collect these samples from many hospitals of the Spanish territory. What we do with this dried blood spot is, in first attempt, the determination of the specific activity of the enzyme through a fluorimetric assay, and this is always often useful to establish a definitive diagnosis for men. Afterwards, we may confirm the diagnosis by checking the mutation in the GLA gene, and this step is compulsory for the diagnosis of female who have a reduced activity of the enzyme and not a null residual activity. It may be strange that we are looking for female patient in an X-linked disease. Actually, what happens here, it has been reported that the X chromosome has an aleatory, random epigenetic inactivation in the different cell types. So the females may develop a phenotype that maybe includes, that may be a severe phenotype. In the cases that are still uncertain, we also measure transcript level of the GLA gene and can examine the biopsy. So in the last 5 years, we received up to 662 samples and identified 72 Fabry patients. The samples were coming from different medical departments. And the positive patients basically belong to 15 families and 8 sporadic cases. As you can see, there is no frequent mutation for this disease. Basically, every family has its own mutation. In the total account of patients, I didn't include the 3 with the D313Y mutation, since this aminoacidic substitution is still controverted in the field. Many experts believe that this is just a polymorphism, since based on studies that have been performed in vitro with the cloned protein. But many other experts believe this is indeed a mutation, since all the patients have a considerable reduction of the enzyme activity and also a clinical phenotype. This is the hugest family we have characterized. We find 11 female patients with an heterozygote mutation and 8 males in the same family. What I want to point out is that we also found between our patients up to 40 of them who just had an alteration of the nucleotide sequence in the intronic regions. And in particular, we found a few SNPs and a little deletion in the intronic region near to the exon 3 that has not been validated as a polymorphism, and we think it's worth to further characterize. I'm showing this because, and also in these SNPs are often present individually, but also as an association of 2, 3, or 4 SNP at the same time. We are showing this because we have founded reason to believe, as we can see later on in the presentation, that sometimes the association, this polymorphism, may act actually against the transcription of the gene or be involved in some kind of epigenetic modification. And so we wanted to further characterize all these patients. Moreover, we also performed 2 screenings for Fabry disease in patients with renal failure from the hospital of Donostia, and another screening in patients with multiple sclerosis from the hospital of Saragozza Lozano Blesa. This second option is not obvious, of course, but we had this idea because indeed sometimes a Fabry patient with neurologic symptoms are misdiagnosed with multiple sclerosis due to the similarity of the MRI scanning. Also, we have to say that all the Fabry patients detected in this cohort didn't show oligoclonal band, neither spinal cord involvement. So the result of the screening were, when for the first screening, we see that we couldn't find any patient with a clear diagnosis of Fabry disease, but we do find a female heterozygote for the controverted polymorphism D313Y. And in the second screening, the one of multiple sclerosis cohort, it was, we analyzed 154 patients with an age range of about 40 years. And also in this case, we didn't have a clear Fabry patient, but we did find a bunch of females, 8 to be correct, who had a considerably decreased activity of about 50% reduction or even more, who present the intronic SNPs I was talking about earlier, and also the uncharacterized deletion, and association of this polymorphism. So we are now in the process to see if somehow this nucleotide sequence alteration can be involved in Fabry phenotype. I'm so insisting with this intronic change because actually we have already some result in this sense. In fact, this is the case of these two patients. He is a male diagnosed of Fabry disease through the enzymatic assay, who have a really severe phenotype with neurological implication, and his daughter of 7-year-old who started also to have some symptoms. In both of these patients, we just found the intronic SNPs in the regions close to exon 1 and exon 4. Characterizing the transcript levels, the GLA transcript levels in those two patients, we found that there is a significant reduction of the GLA transcript in both patients compared to individual controls. To confirm the data, we also checked for GB3 deposit in tissue in a skin biopsy from the little girl. And we did find a few dermal fibroblasts with GB3 deposited. And apparently also the expression of alpha-galactosidase A in tissue seems to be decreased. The skin biopsy actually is, we think, is a very useful tool for diagnosis of Fabry disease in uncertain cases. In fact, this is a very little invasive technique that allow us to study many different cellular structure, tissular structure. Of course, the dermal fibroblasts, but also blood vessels, smooth muscle, eccrine glands, epidermal nerve endings, and concerning the epidermal nerve endings, I want to say that it has been reported that in Fabry patients there is a significant reduction in the number of epidermal nerve endings crossing the dermoepidermal junction. So calculating this number may serve as an index for the neuropathic involvement in this patient. Concerning the second objective of our project, we are interested in the pathogenesis of Fabry disease. The most obvious hypothesis is, of course, that the deposit itself leads to cell rupture. But we now know that there are many factors affecting the pathophysiology of the disease, that for sure there is an inflammatory processing coacting with the other mechanism. In fact, leukocyte function is impaired. Also, that GB3 itself is identical to the CD77, which is expressed in the membrane and has been related with apoptosis and necrosis. GB3 accumulation may also stimulate ROS production. And we know that the ischemic vascular events occurring in Fabry disease are caused by the swollen endothelial cells, and of course an inflammatory component has to contribute in this step. So our hypothesis that, coming back to the project, we have an hypothesis that is still to be proved. We believe that maybe some kind of involvement of the purinergic signaling pathway in Fabry disease. This signaling pathway has been involved in different processes, including the initiation of the inflammation, T-cell activation, and most of all, they participated in the in different sensory systems, especially in the one affecting pain, manifesting pain. Also, that they are expressed in the microglia and cause oligodendrocyte damage, contributing to form lesions similar to those observed in Fabry patients. and they also control autophagy. So for those of you who are not familiar with this class, with the signaling pathway, just say that the purinergic signaling is triggered by this receptor when they are stimulated with ATP, and ATP is the ligand of the receptor in this case. There are two classes of different receptors. In this case, the ones that are more willing to participate in Fabry disease are the P2X receptors, which are basically ion channel, selective to cations, but who have the peculiarity to allow also the passage of little metabolites. What we have did to prove our hypothesis, at the moment very little experiments, but we are working hard and we will keep on working on it. So we just basically checked the basal expression of the P2X receptors in monocytes derived from Fabry patients. We use monocytes because they are easily available and they are similar in a certain extent to the microglia. We apparently found downregulation of the receptor. Of course, the statistic of the experiment should be ameliorated, and also it will be necessary to perform more experiments upon stimulation of the receptor. We still cannot know if this may be a primary effect or more likely a consequence of the disease in this patient. I also said that the purinergic factor pathway controls autophagy, and we made some autophagy study in a patient with Fabry disease. We analyzed the myocardial tissue of one female patient, either zygote for the Q279R mutation. She, of course, has a cardiomyocyte full of GB3 deposit, and also apparently there is an increase of the signal of LC3, which is an autophagy marker, and the autophagic process is also appeared to be confirmed in the study of the ultrastructural study of the myocardial biopsy. We wanted to check if this is result by Western blot, but unfortunately the amount of tissue was a little bit too small to check it directly on the myocardial tissue. So I used what I had available, that were fibroblasts derived from a skin biopsy of the affected son of this lady. And we see that there is an increase in LC3 expression even upon stimulation with ATP of the receptor when compared with the control. So, and finally, to almost conclude, the third objective I'm interested in, since I've been working this previously during my postdoc, will be to build up a construct based on adeno-associated virus to possibly transduce cells of Fabry patient. In particular, what we want to do is to transfer cells direct in the kidney, since the construct that has been made up to now are not very well expressed in the kidney. So we have a strategy for this. And we are planning, if possible, to do the cloning and production phase in collaboration with one of the tutorial centers of the BioCaps project through one of the tuning activities. Just to summarize and conclude, say that the screening of the enzyme activity and the genetic sequence of GLA in group of patients with a clinical suspicion may really facilitate early diagnosis. Also, that it is worth to look at the intronic change in the sequence, since they may be affecting some transcription pathway or be involved in some kind of epigenetic modification and develop the phenotype. The skin biopsy may be a useful tool to confirm diagnosis in doubtful cases, which may be, for instance, a female patient with a residual activity and no mutation in the GLA, but just in the intronic sequence. And also that the pathophysiology of the disease is still mostly undetermined, but we have founded research to suspect that this pathway may be implicated in the physiopathology of the disease. And thank you to all the members of the neuroscience group at the IBIV and to all the physicians that are actively collaborating with us. And to the sponsor and to all the audience, of course. Yeah.